Okay, so last time when we left off in our virus lecture, we were looking at this slide here, which showed the different pathways that can be taken for our replication of RNA viruses. If you're a single-stranded plus sense RNA virus on the top, a negative sense single-stranded RNA virus in the middle, and then finally double-stranded RNA virus on the bottom. And so the artist has used this color coding to indicate intact viral genome and then the plus sense strand or the minus sense strand. And so we're going to look at each one of these in turn to kind of walk you through it. So starting with the one up here on top, shown on the next slide. And so here we see a virus capsid. This looks like one of those icosahedral capsids or polyhedral capsids, and the RNA genome inside. And after the virus has attached to and penetrated or entered into the host cell, it will uncoat and thereby release the viral genome as well as any packaged viral proteins, for example. So at this point, the viral genome, because it is plus sense, it is essentially identical to a messenger RNA transcript, or at least it mimics one very, very closely. And so if I jump ahead to the slide that I pulled from Chapter 8, if we look closely, just to remind you what a messenger RNA molecule looks like, let's take a look here at a transcription kind of zoomed in close. And if we take a look at this transcription, we see here's a template strand of DNA, and here's a strand of RNA being synthesized that's complementary to the DNA. So G is pairing opposite C, A opposite T, and then most important to realize is that because this is RNA, there will be a substitution of uracil for thymine opposite the A. Okay, this is a very close mimic of the thymine. Notice that the synthesis appears to be proceeding from left to right, and because we know that synthesis always goes in the 5' to 3' direction, whether or not you're talking about RNA or DNA, we would then label this side of the RNA molecule as the 5' and this side as the 3'. And so then the DNA molecule obviously must be the exact opposite, with the 5' being over on this side and the 3' being over on this side. Now, imagine if we just took this transcript RNA, this messenger RNA, and pulled it out of the slide and put it into this one and packaged it inside of a capsid. That's more or less what a plus sense RNA genome looks like. Again, it's a very good mimic for a messenger RNA molecule. So good, in fact, that this molecule can be recognized in the host and translated into proteins. And so it's recognized by the ribosomes, and proteins can be made directly from the viral genome. What does that mean? Well, later on, we're going to obviously need to make a complementary strand to this plus sense RNA in order to make more of the viral genome, which is plus sense. Okay, and so we need to make a template from this RNA molecule to make more copies of this RNA molecule. And so what we're talking about is basically making RNA from RNA. And as we discussed in class, that's not something your cells typically do. We make RNA from DNA typically, and so our RNA polymerases, the ones in our cells, are designed to recognize DNA as a template, not RNA. And so what the virus needs is an RNA-dependent RNA polymerase, one that uses RNA as its template. And so these plus sense viruses have an advantage in that because their genome itself can be recognized by a ribosome as instructions for making a protein, the virus is not forced to bring with its own RNA polymerase. In many cases it does, but it's not required to, because you could have a gene on this quote-unquote messenger RNA, that is the viral genome, that could code for your RNA-dependent RNA polymerase. And so once the ribosome would recognize this molecule and translate from it, it could thereby synthesize your RNA-dependent RNA polymerase. In any case, whether or not you force your host cell to make your RDRP, or you package it along in your capsid and release it when you uncoat inside the cytoplasm, at some point you'll be synthesizing RNA from RNA, and that's what the artist is showing here. And so here's the plus sense stranded genome, and it is being transcribed into a minus sense strand of RNA. And now this minus sense strand of RNA down here can now be used as a template to make more plus sense. So we're just flipping back and forth. We're starting with plus sense, making a minus sense strand, and then using that minus sense to make more plus sense. And again, you need this minus sense template in order to make more copies of this, because this is the name of the game if you're a positive sense RNA virus, because you need copies of this to put into your capsids to make your progeny virus. And so also on this genome over here are going to be your plus sense genes, for different proteins like the capsid protein. And so late in the replication cycle, you'll see these capsid proteins being synthesized off of these plus strand RNAs. And again, that's going to be mediated by the ribosomes. So if you put together the plus sense genome with the capsids getting assembled into with the capsid proteins being assembled into capsids, you add all that together and you're going to have maturation of mature virus particles, which are eventually released from the host cell, either by budding or lysis of the cell. Now, if we go forward and look at negative sense RNA viruses, it's a little bit of a different story. Again, you have attachment and entry into the cell and uncoding. But this time it's critical that the virus brings with its own RDRP. Again, what we're talking about is an RNA-dependent RNA polymerase. And the reason why is because this viral genome, in this case, is a negative sense viral genome. And so it cannot be recognized by a ribosome and direct translation. Instead, this needs to be transcribed into a plus strand RNA in order to make any sort of proteins. And the artist or this diagram indicates that down here. And so you see here that the plus strand RNA must first be transcribed from the negative sense viral genome before any proteins can be synthesized. And that process is being shown here. Now, once you have these plus sense RNAs that were made, yes, indeed, those can then be serve as messenger RNAs that can then be translated into proteins, and they can make capsid proteins as shown over here. These plus strand RNAs also have another function, and as you may have anticipated, that function is that they serve as templates for additional copies of the negative sense RNA genome, and that's what the artist is showing down here. So now if we take these capsid proteins as well as the newly synthesized negative sense strands of RNA and we add all that together, you're going to have the formation of complete nucleocapsids or virus particles, and those will be matured through different processes to assemble mature virus particles. And again, they can be released from the host cell, typically by budding, but in some cases by lysis. Now, if we put together those previous two stories of the plus sense virus and the minus sense virus, you more or less have the story of a double-stranded RNA virus. So again, if we zoom in a little bit here, and let me move the slide over a bit, you see attachment and entry of the virus and uncoding, and then you have this viral protein here also that may have been released during the uncoding process. And so depending on the virus, you may have that your plus sense strand can be immediately translated and recognized by a ribosome, but more likely is that it's a little bit more similar to a negative sense single-stranded virus in that mRNA is going to be produced as a result of an RNA-dependent RNA polymerase that the virus brought along with it. During the previous replication cycle, an RDRP was incorporated into the capsid, and that's released, and that RDRP is going to direct the synthesis of both plus sense and minus sense strands. Now, one thing that should be noted, it says here, mRNA is produced inside the capsid and released into the cytoplasm of the host. So it turns out that before the encoding process has taken place, we actually have buried inside the capsid that copy of RDRP that I just mentioned. In fact, entry into the host cell is going to stimulate the virus to start producing messenger RNA before the capsid is completely uncoded. And so now we have messenger RNA transcripts that can be used to make virus proteins. And again, the positive sense strands are going to serve as templates for the negative sense strands and vice versa. And so you see here RNA polymerase initiates the production of negative sense strands. The mRNA and negative strands form the double-stranded RNA that's incorporated into the new viral genome. So you add those plus sense and minus sense strands together, and now you've recreated your double-stranded RNA genome. And indeed, when all that stuff assembles during the maturation process, let me go back to the slide here, you can then make your mature virus particle as shown up here that again can leave the host cell by either budding or lysis. Now let's take another example, and this time we'll talk about multiplication of a retrovirus. And you've heard of retroviruses, I'm sure, before in reading or just in the newspaper. And retroviruses have been associated with humans for a very, very long time. And one of them is responsible for one of the biggest pandemics in human history, and that is the HIV pandemic that really started to gain steam back in the 80s. And so let's take a look at a retrovirus replication cycle. And so here we see a host cell, and the virus is entering by endocytosis. And so this is an enveloped virus, so you can see there's an envelope around it, and the spike proteins of the virus are embedded in that envelope. And after attachment and entry into the host cell, the virus is going to partially uncoat, and that's what I've added this note here. And actually, while the capsid is still somewhat intact, a very important enzyme is going to play a role. And what that enzyme is reverse transcriptase. And that's shown here very tiny inside the capsid. But there's an enzyme that's packaged in the virus capsid, and this reverse transcriptase is going to reverse transcribe RNA back into DNA. And so this was considered heresy when it was first suggested that we could actually make DNA from RNA, but it was later proven to be true experimentally through numerous experiments, and resulted in the Nobel Prize for the people who discovered it. And so here we have the viral DNA, and we should note that this is a plus sense single strand of RNA, and the virus actually encodes or contains two copies. They're identical. So please note that these are not base paired opposite each other, because to base pair the strands would need to be complementary. So complementary sequences, but different sequences. In this case, the two sequences are identical, so they're not base paired together. Instead, we have the single strand of RNA now that's been released during the encoding process, and reverse transcriptase is going to synthesize off that a viral DNA molecule, and it will actually carry out a second strand synthesis, so you now have a double-stranded molecule of DNA that is the same sequence as this viral RNA, again substituting T for U. So what's so magical about making a DNA molecule for this virus? Well, by making a DNA molecule, this virus now, will have the ability, if I could scroll down without changing slides. Let's try this. Nope, that's not working. Go back here. Oh, there we go. This piece of viral DNA now has the ability to integrate into the chromosome of the host cell, and there is an enzyme called integrase that is important for that catalyzing that activity. So if we take this piece of viral DNA and essentially insert it or integrate it into a host cell chromosome, the virus now has this tremendous advantage in that every time the cell replicates, this provirus, as we term it, will be replicated as well. And so the virus really needs to do nothing to replicate itself other than just stick around inside the host cell chromosome. And there it's perfectly protected from the host immune system, in terms of it doesn't have to enter into the nasty world of where it could be exposed to, say, antibodies or phagocytes. And so at some point, however, the provirus may be transcribed even while it's sitting inside the host cell genome. And when it's transcribed, that will produce new copies of its RNA genome as well as some viral proteins. And in fact, stimulation of transcription is one of those things that may be a result of, say, stress on the host cell, for example. But as these viral proteins are processed and as copies of the RNA genome are made, they can then be essentially packaged into mature retrovirus particles that then can bud out from the host cell and acquire their new envelopes as little pieces of plasma membrane. You may notice that there are these little spike proteins on the surface, and again, those are very important for attachment and entry into the next host cell. And what happened was when proviral genes were being transcribed and translated into proteins, these little spike proteins are synthesized and they're actually transported onto little islands on the host cell plasma membrane, and the virus will then acquire those spike proteins as it acquires the host cell plasma membrane to then use as its own envelope. So one thing that might interest you is that we did talk about in a few earlier slides how viruses are detected. Traditionally, it's by essentially trying to take a patient sample and infect cell cultures to see if the cells then become sick or infected with the virus. But more recently, we've been developing genotypic methods to detect viruses in patient samples. And so these are some data that I actually collected off of patient samples down at Gunderson Health in La Crosse, in the laboratory where I work down there. And what we can see is that this is the output of what's called a quantitative or real-time PCR machine, in which we're looking for production of pieces of DNA that were synthesized off of copies of the viral genome. And so basically, the earlier that you see this curve deflecting, the more virus was in that patient sample. And after we essentially produce copies of portions of the viral DNA, we can then determine at what temperature do they melt, and depending on the melt temperature, we can determine whether the patient was infected with herpes simplex virus 1 or herpes simplex virus 2. And so I just wanted to share with you some of the data that illustrate the way we now try to detect viruses much more quickly. Now, HSV is actually a DNA virus for the Zika virus outbreak that's currently ongoing. A very similar test is being carried out, but before the amplification of Zika virus DNA can take place, we actually need to use an enzyme that we borrowed from a retrovirus. This reverse transcriptase you can now buy and use in just a test tube type of reaction. We use the reverse transcriptase to reverse transcribe Zika virus RNA genomes into DNA complementary strands that we can then amplify up with our PCR reactions. So this is just a nice summary of what we just discussed, and so I recommend that you review this and be able to understand the differences between the different types of virus replication. Now, your book also has an interesting section on cancer and viruses, and so I'd like to spend a few moments talking about that. So it turns out that about 10% of all cancers are induced by virus infections, and this includes nearly all cervical and anal cancers. And so the way that viruses can induce cancer is by integrating their viral genes into the host cell DNA, very similar to what we just discussed with the retroviruses. And so what's not shown in the slide, but what I'd like to mention is that cancer basically is a disruption of the cell cycle, so that the cell keeps dividing without any inhibition, and that results in the formation of a tumor. And then if that tumor portions of that tumor break off and circulate throughout the host patient and then establish tumors at other sites. That process is called metastasis. And so it's really those two events of tumorigenesis, meaning the creation of tumors, and the metastasis, the spreading of these tumors to other locations, that we use to define something as a cancer rather than, say, just a benign tumor. So how do we disrupt the cell cycle? Well, basically, the analogy that I like to give is think that you're driving a car. And to control the momentum or the speed of the car, you essentially have two things to work with. You have a gas pedal or accelerator, and you have a brake pedal or a decelerator, if you want to say. And so cells, to control their cell cycle, have a similar type of control. They have some genes that are in some ways proto-oncogenes, in which they can promote division or entry of the cell into the cell division cycle, so we'll keep dividing. And think of those genes or gene products as the gas pedals. And then there are other genes that are considered suppressor genes that will essentially act as a brake to stop cell division or uncontrolled cell division. And so if you have a mutation in either one of those types of genes, that could then result in making you more susceptible for developing a cancer. So imagine, again, since cancer, we're talking about uncontrolled cell division. You're talking about either sticking the gas pedal in the down position so that the car keeps going forward or the cell keeps dividing, or say cutting the brake line so you can no longer hit the brakes. And so when we talk about oncogenic viruses that cause cancer, it's viruses that contain genes that are either like a stuck gas pedal or a broken brake line type of interaction with the host cell. And so when a cell is infected with one of those oncogenic viruses, it becomes what we call transformed. And these transformed cells will keep growing and they lose what's called contact inhibition. So even if they're all crowded into a culture or into a tissue, and whereas most cells will respond with that crowding by stopping their cell division, these transformed cells will just keep dividing and pile up on top of each other, thereby forming a tumor. And so there are examples of both DNA and RNA viruses that are oncogenic, that are capable of creating cancers. But again, what's in common with all of these is the fact that you typically will see integration of the viral genes into the host cell genome. So now I'd like to talk a little bit about latent versus persistent viral infections. And so what we're talking about with this is basically the titer or concentration of viruses over time during an infection. And so if we look at a chart, this is number of virions over time, starting out with a relatively short period of time of days and then longer periods of time of months or years. We can see that a viral infection will typically have a curve that looks something like this, in which you'll have, say, your introduction of live virus into the host, and that virus becomes relatively quickly undetectable as it enters into host cells and starts replicating. So you don't have any mature virus particles outside of the infected cells. Then after that time of lack of detection, the virus often is replicating quite rapidly and being released into the host's say bloodstream, and that's the phase of acute infection where typically you see the acute symptoms in infected patients. It's not uncommon for some viruses, such as the ones that cause cold sores or shingles, for example, to then go into a long period of what we call latency, in which the virus is not actively dividing inside the host. Now please ignore this blue line for the moment, because we're talking about latency and not persistent infections. And so you notice that there's actually this gap between the red line and this green line over here, in which you really don't see any detectable virus. And that's what we're talking about with latency. And so typically years later, perhaps, there could be a stress on the host, lack of sleep, exhaustion, whatever. If you think about cold sores, that's when people are tending to have their cold sores erupt again, and you'll have the viruses then triggered to start actively replicating so it can be transmitted to a new host, and then you'll see a new blip of virus showing up, and that's indicating the recurrence of the infection, or the recurrence of the virus replication, I should say, that's indicative of a latent infection. Now, in contrast, there are other infections that are called persistent virus infections, in which there is, say, a very low level of replication over a number of years until later on the patient is compromised enough that the virus titer rises rapidly, and these infections, unfortunately, are usually fatal. And so it was only very recently that we have accumulated the data to show that AIDS is actually an example of a persistent infection and not a latent infection. Because as you're probably aware, there'll be a period of acute infection, usually within the first couple of weeks of acquiring the HIV virus, at which point the patient will then resume kind of a normal appearance and feel fine. And it was over the course then of years that there'll be a slow replication of the HIV virus in that patient that then later, when the patient is debilitated enough, will result in the development of full-blown AIDS, which untreated is invariably fatal. So then, moving on, finishing up, I'd like to talk about a couple of other topics. Prions. Now, prions this stands for proteinaceous, meaning they're made of protein, infectious particle. Basically, what we're talking about are infectious proteins. So there's no nucleic acid associated with them. And again, this was considered heresy when it was first suggested, but later resulted in a Nobel Prize when it was proven. And examples of diseases that are caused by prions would be the spongiform encephalopathies, sheep scrapie, Creutzfeldt-Jakob disease, etc. And these result in usually severe neurological symptoms that typically are invariably fatal unless the patient, are typically fatal because there's really no treatment for a prion disease. And so again, these are not viruses; they're proteins. And what you should understand if you look at prions is that these proteins are actually normally found inside cells, but they're in a form or a shape in which they're harmless. They're actually necessary for the cell, in this case, neurological tissue, for healthy neurological tissue. And what happens is that the protein can sometimes misfold into it has the exact same sequence, but has a different shape. And this misfolded protein will accumulate in brain cells and form these plaques that result ultimately in the death of the cell. It may be an indirect result, but the cell will die with continued plaque formation. And so here we have an example of a normal cellular type of prion protein being produced by the cell and secreted onto the surface. And then as a result of exposure, say, to the scrapie form, the misfolded form of this protein, it might be introduced into the patient, and it will react with the normal form and essentially induce it to acquire that misshapen conformation. And so that's the process that you see here and here, in which this normal shaped protein now is changed into this misshapen one. And so that it can result in essentially a cascade of events, a chain reaction, in which more and more of the scrapie form of the prion protein is produced. And these scrapie forms are recognized as being that there's something wrong with them, and the cell actually tries to destroy them through endocytosis, but instead of them being destroyed by lysosomes, they are accumulating in endosomes, and eventually, when they're trying to be destroyed through fusion of the lysosome and endosome, typically results in cell death because they're more or less indigestible. And so very dangerous disease, and as we mentioned, prions cannot be destroyed by autoclaving, but only by harsh chemical treatments such as the use of proteases or acid-base hydrolysis that might work. And then finally, we'll wrap up talking about viroids and plant viruses. And so plant viruses have the challenge of trying to infect a cell that's surrounded by a cell wall. And so typically plant viruses are only transmitted through injuries, that the plant tissue is damaged. So a classic way for a plant virologist to infect host plants is to abrade the leaf with a piece of sandpaper, and then just use an eyedropper to drop a suspension of the virus onto the plant leaf. Another way that plant viruses can be transmitted directly into cells is through the feeding damage of certain types of insects that actually act as vectors for the virus. So that's plant viruses. Viroids are only known from plants, and they're kind of like the opposite case of a prion. So a prion is an infectious piece of protein. In this case, viroids are, again, not viruses, but they're infectious RNAs. And so these are RNAs that can be introduced into host cells and result in disease because they are self-replicating inside the host. And we'll close with a discussion. I have a bunch of slides that talk about different types of viruses and their characteristics, and I won't bore you by reading through these slides, but what I want to point out is that this is the level of information that you'll be held responsible for in terms of knowing the different kinds of viruses and what diseases they might be associated with. So be sure that you examine these slides of the various virus families. So that's it for the virus lecture, and I wish you the best of luck in your studying. And again, either tonight or Saturday morning, I'll post a discussion section to D2L so you could ask some questions on this material or anything from chapters 8 and 13 prior to Monday's exam. That's all for now. This is Casey Finnerty from the Biology Department at Winona State University.